Once the action potential has traveled down one neuron's axon, it comes to the synapse, a place where one neuron gets very close to but doesn't quite touch part of another neuron. The electrical signal can't directly jump this gap. Instead, a chemical signal is used to convey information about an action potential in the first, or presynaptic neuron, to the next, postsynaptic neuron. In fact, each axon will form synapses with many different downstream neurons, so a single axon may be presynaptic to thousands of postsynaptic neurons. The arrival of an action potential at the synapses causes the release of a chemical, called a neurotransmitter, by the presynaptic neuron into the synaptic cleft, that gap between one neuron and the next. The neurotransmitter diffuses across the gap, like a drop of dye in water, and then binds to specialized receptor proteins embedded in the postsynaptic membrane. That receptor then conveys a signal to the postsynaptic neuron. Let's talk a little more about neurotransmitters, those chemicals that are released from one neuron onto another at a synapse. The brain uses dozens of different neurotransmitters with a wide array of chemical structures. Some are small molecules. Many of them are closely related to the amino acids that are the building blocks used to make all proteins. These are generally manufactured from their chemical precursors through the actions of one or more enzymes in the cell. Others are peptides, short amino acid chains. Some are lipid-derived, and yet others are gaseous molecules. Some neurotransmitters you may have heard of include dopamine, serotonin, and epinephrine, or adrenaline. Each neuron uses the same neurotransmitter, or a combination of neurotransmitters, at all outgoing synapses. Neurons will sometimes be classified or described based on which neurotransmitter they use. For example, a dopaminergic neuron is one that releases dopamine at its synapses. These neurotransmitters are generally stored inside synaptic vesicles, little spheres of plasma membrane inside the neuron, which get transported to the synapses. When an action potential in the presynaptic cell travels down the axon and arrives at a synaptic terminal, the change in membrane voltage causes voltage-gated calcium channels in the cell membrane at the synapse to open, allowing calcium to flow into the neuron. These calcium ions then interact with specialized proteins on the cell membrane and on the synaptic vesicles filled with neurotransmitter, causing that synaptic vesicle to fuse with the cell membrane and dump its contents outside the neuron into the synaptic cleft. Botulism and tetanus are both caused by toxins that disrupt the molecular machinery responsible for vesicular fusion and neurotransmitter release at the synapse. Now, the neurotransmitter diffuses across the synaptic cleft and binds to the receptor protein embedded in the postsynaptic membrane. The receptor will be matched to the neurotransmitter being released at that synapse. The effect of the binding of the neurotransmitter to the receptor on the postsynaptic neuron varies depending on the particular neurotransmitter and receptor, and that'll be the subject of our next video.